Today we're going to continue talking about bacteria and archaea. You'll see that in the slides, they go through different parts, different groupings of bacteria. All right, we are not going to cover those in class. You are responsible for them. All right, so part of that's going to be a review from what you did in lab, right, with your dichotomous keys and classifications of bacteria. All right, you are responsible for knowing proteobacteria, spirochetes, et cetera, and examples of them and main characteristics of them. All right, most of that is in the slides. I'm not going to go through it. It's sort of pointless and sort of repetitive for me to go through it in lecture, where you can just study it and you can understand and know it for that purpose. Understood? All right, so what's on the slides of the bacteria that I expect you to be able to identify, right? So you know what proteobacteria, what they look like cyanobacteria, what their characteristics are, etc. Okay? So that's first off. So you'll see we'll get to that in a little bit. First, we want to talk about, we left off talking about characteristics of prokaryotes that allow them to evolve quickly, right? To adapt to their different environments. We talked about their different metabolisms, right? How they obtain energy, right? A chemotroph versus a heterotroph versus an autotroph, photoautotroph versus a chemoautotroph, et cetera, et cetera, et cetera. Okay? And helping to allow for that adaptation, that evolution, is the idea or the ability of bacteria to undergo genetic recombination. All right? Genetic recombination is the ability to combine DNA from two different sources. All right? So, in terms of prokaryotic, in terms of bacteria, we've already went over a process of genetic recombination. What would that be? It's covered in last chapter. Give me an example of genetic recombination that you see in bacteria. What's that? Lysogenic phase, right? Infection by a bacteriophage, bacteriophage that undergoes the lysogenic cycle, injects the DNA. That DNA gets incorporated into the chromosome as a prophage. That is a genetic recombination event that occurs. All right? So, what you end up seeing is that bacteria can perform this genetic recombination in three distinct ways. Transformation, which you did in lab, right? Picking up pieces of DNA from the environment. Okay? Transduction, which is a transfer of DNA through virus infection. All right, so a bacteriophage. Bacteria can be transduced through bacteriophage infection. And then conjugation, right, where two bacteria link together through a sex pilus, that little tunnel, and there's an exchange of genetic information that way. Okay? And so movement of genes among individuals from one to another, part of a population, is horizontal gene transfer. Right? What is vertical gene transfer? Right, so parent cell to daughter cells, right, or in that lineage, where you have two separate populations, okay, or two separate organisms, and they transfer genes to one another. This is horizontal gene transfer. All right, so we go through transformation with pickup, which is defined as picking up DNA from the surrounding environment. Cells die, all right, they leave remnants of DNA that may get picked up by nearby bacteria. And transduction is the movement of genes between bacteria by bacteriophages. All right? And you'll see this in a second. What occurs if during the lytic cycle or the lysogenic cycle, let's say the lysogenic cycle of a bacteriophage, whereby the chromosomal DNA gets broken down in the preparation for release of new virus. What happens if the DNA genome for that virus, the viral genome that's been replicated, doesn't get packaged into the virus, but instead a set of bacterial genes does? Then that virus goes out and infects another cell. But it's not going to be infective in that it's not going to pass on the viral genome. It's going to pass on what? That bacterial genome. And then that genome gets incorporated into the infected bacteria, and now that ends up being a transfer of genes. Okay? So bacteriophage can pass on not only the viral genome, but whatever bits of stuff that gets incorporated from the previous infected cell onto the next cell. And that is the idea of transduction. Right? So here's basically a schematic of that. Okay? Phage DNA incorporates in the donor cell. You can see they're using A and B as showing where these genes are. All right? The phage DNA is replicated. You know this cycle. This is just the lytic cycle. Okay? But the difference being is that a piece of the bacterial DNA are there in step 3. Okay? Right here. Piece of the bacterial DNA is incorporated into the virus instead of the viral genome. Okay? That gets then transduced into the next bacterial cell. Crossing over event occurs because they are like cells, and that's similar structure DNA. Right? Crossing over event occurs, and that piece of DNA then gets incorporated into the cell. Okay? If you are saying that this first, in step 1, that first cell is, say, positive gene expression for A, okay, and this new infected cell doesn't have A, then what's going to happen to this newly infected cell? It's going to have a new gene, right? We think about that in terms of antibiotic resistance. We say this is a susceptible gene or susceptible cell at step 4, right? And the gene for antibiotic resistance for a certain antibiotic is gene A, and it's going to be transferred from gene A from the cell in step 1 to the cell in step 4. Now that recombinant that you see in step 5, okay, is an antibiotic resistant, right, as an example. Okay, but it's a way of moving genes from one cell to another, horizontal gene transfer, transduction. Okay. Conjugation and plasmids. Conjugation is the process where genetic material is transferred between prokaryotic cells. So transformation is picking up DNA from the environment. Transduction is a transfer of DNA via a virus, via a phage. Okay? And conjugation is one to another. One prokaryotic cell directly to another prokaryotic cell. All right? Transfers one way, for the most part, right? And in E. coli, as an example, you end up with the actual linkage between two cells through the sphere. Okay, so you have one E. coli cell, right? Then there's an extension of the sex pilus. They conjugate, they come together, and there's a transfer of genetic material from one to the other. Questions on those? All right. Plasmids. What are plasmids? What's that, Abby? Circular pieces of DNA. Are they part of the chromosome or not? No, they're extra chromosomal circular pieces of DNA. But they are transcribed, and they have genes, they may have genes on them. Okay? And we're going to look at one particular plasmid. And you did plasmids in lab. You introduced a plasmid into bacteria via transformation, which was then transcribed and allowed certain cells to grow. And you ended up with happy, glowing GFP colonies, right? Unless you screwed it up like Nick. Well, that's good, though. Did something right. All right, so the F factor is an example. All right? So the F factor is a plasmid, is a piece of DNA, which is required for the production of this sex pilus. So that sex pilus, all right, is actually a gene in the cell. It's produced by a gene in the cell that allow it to produce this. Okay? So you have an F factor. Cells containing the F plasmid are said to be F plus. Right? This also, F stands for fertility. Okay? And they functions as DNA donors during conjugation. Right? So an F plus cell can produce a sex pilus and will transfer DNA from that cell to another cell. All right? Cells without the F factor, right? They are your DNA recipients. In bacteria, DNA exchange is one way. So always going from F plus to F minus. Okay? And when this occurs, the F factor is transferable. So what's going to happen to the recipient cell after conjugation? So the F plasmid is going to be transferred to it. It's going to become an F plus cell. Okay? So here's your F plus cell at the top. This is F plus. Here's your F minus cell. Okay? What's going to occur is that the mating bridge, that sex pilus, has been produced by the F plasmid. You get replication of the F plasmid, and then it transfers through the sex pilus, the mating bridge, into the F minus. All right, so your donor recipient synthesized complementary DNA strands ends up with now 2 F positive cells. Yes.
So is the only thing that gets transferred just the F plus?
Just the plasmid.
Just the plasmid, so there's no other like genes in the plasmid?
Well, whatever genes are on the plasmid.
Okay.
All right, so it's just the F, that whole plasmid. It might be 30,000 base pairs. It might be 65,000 base pairs. And whatever is on that plasmid will go from one cell to another. So in terms of research in the lab, you could engineer a plasmid, an F plasmid, that has an antibiotic resistance gene on it, and whatever gene you want to look at, and infect one cell, and then it will, by its biology, infect the other cells. And that gene would be transferred on. Okay? Questions on that? All right? The F factor can also be integrated into the chromosome, similar to a prophage. Okay? A cell with, once it gets integrated into the chromosome, what's going to happen in terms of cellular reproduction? When the chromosome replicates, what's going to happen? The F factor is going to replicate on it as well and be passed on to the next cell. All right? So and then that can function as a donor, all right? And then that you end up with DNA from. Okay? Other plasmids in terms of importance. All right? Yes, Emily.
So plasmids are replicated with the cell?
Yep.
Or is it integrated into the DNA?
The plasmids, most plasmids are structured in a certain way, okay? So that most plasmids have, say, in terms of structure, they're circular DNA, right? And they'll have certain genes on it. Say this might be the gene for the gene A. Here's an antibiotic resistance gene, right? And say this gene is the one that your favorite gene, whatever it might be, okay? Most and all plasmids also have an origin. Okay? So they all have the ability to replicate. They have the spots and the structure to replicate as the cell is replicating the DNA. So it doesn't have to get integrated into the genome to be replicated. When DNA is replicated, it's also replicated.
So then why is it important that the F factor can also be integrated into the chromosome itself?
In some cases, the plasmid can be kicked out.
Okay.
Okay, so bacteria can sort of incorporate plasmids and also expel plasmids. So once it gets incorporated in the genome, it's sort of stuck there.
More permanent.
Yeah.
Thank you.
All right, so when we look at the plasmid, if I can make this bigger, there we go. We look at our plasmid again down here. I'll make this smaller for now. We'll look at our plasmid over here. It has a antibiotic resistance gene on it. Okay? What is that going to do for any cell that has incorporated that plasmid? It's now going to be resistant to whatever antibiotic that is. Okay? So if you have, a gene for a beta-lactamase, which breaks down penicillin, that cell now is resistant to ampicillin, and you can use that as a selection tool. All right. So you have your antibiotics, kill sensitive bacteria, but not bacteria with the specific resistant plasmids, right? And then you have natural selection, right? Resistance gene. What do we use for an example of resistance on Monday? MRSA, right? Methicillin resistant Staph aureus, right? And they're becoming more common. Why are antibiotic resistant bacteria becoming more common? What's one reason? Overuse of antibiotics. The fact that you have a cold and you go to the doctor and he says, here, take this penicillin, it'll help you, whereby the cold is just a virus. It's not going to help you combat the cold. Usually the antibiotic helps prevent any secondary infection, such as a sinus infection that you may get from, the symptoms of a common cold or flu. All right, but it doesn't work up against those. All right, questions on genetic recombination. So transformation, transduction, conjugation, and plasmids and what they do. All right, plasmids in terms of their importance. Okay, so then 24.3 is a rundown of bacteria. 24.4 has this is the rundown of bacteria. We're going to cover the first part of it, and then the specifics of it you're going to cover on your own. Okay? So, because of their evolution, their ability to mutate, grow quickly, and adapt to environments, bacteria have divulged into this, or prokaryotes have divulged into a very diverse lineage, or diverse lineages, from photosynthetic bacteria to non-photosynthetic, chemotrophs, autotrophs, etc. All right, so there's diverse structural and metabolic adaptations that are present. All right, and they inhabit every environment known to support life, and even some that have not been shown until the identification of bacteria there to support life. So, prokaryotic diversity. All right, most of the time what we do to change is molecular systematics, looking at DNA and RNA sequences, structures of proteins, right, to show how bacteria are different from one another, how they react with their environment in terms of their metabolism, what they use. It's all molecular approaches. Okay? And then this also led to the splitting of the kingdom, or the domain, into prokarya, or bacteria, right, and archaea. Both being prokaryotic cells, they have different structures. All right? Archaea have different types, different ribosomes than bacteria do. That was one of the distinctions, all right? Archaea are more closely related to eukaryotic cells in terms of their ribosomes, their gene structure, etc., than bacteria, all right? And so looking at this and the sequences and structure of the molecules also leads to separating and working on the phylogeny, the classification of proteins. So here's sort of a breakdown, and this has been broken, doesn't reproduce very well, but it is what it is. So you have your eukaryotes, and then your, as a domain, you then have your archaea, right? 4 different classes of archaea as a separate domain, and then your 5 classes of bacteria. Proteobacteria being your true bacteria, chlamydias, spirochetes, cyanobacteria, and gram-positive bacteria. Those 5 classes are the classes you're responsible for in terms of understanding the characteristics of bacteria. You do not need to know the characteristics of the 4 classes of archaea. Okay, we're going to go through the general characteristics of the archaea in a little bit. All right, so archaea share certain traits with bacteria and other traits with eukaryotes. So they sort of are the gray area between the two. All right, I already said that the structure of ribosomes and the size of ribosomes is more closely related to eukaryotes than it is to bacteria. But archaea still don't have a membrane-bound nucleus. Okay, so there is some difference between the two. Okay, and that's then your splitting of the 3 domains. Here are your characteristics and comparison between the 3 domains. And you should study this. I got something in my eye there. Nuclear envelope, is it present in bacteria and archaea? No. Membrane enclosed organelles? No. All right. Peptidoglycan cell wall is present in bacteria but not in archaea. Okay. Their cell walls differ. All right. The lipids that are found in their cell membranes are different in archaea than in bacteria and in eukaryotes. All right. Several different kinds of RNA polymerases, whereas bacteria only has 1. Okay? Initiator amino acid for protein synthesis. The structure in bacteria, it's formylmethionine. In eukaryotes and archaea, it's just methionine, right? Which is a different structure. The purpose of formylmethionine in bacteria is that the formal structure actually serves as sort of the 5' cap for the beginning of that. All right, of that initiator sequence. Helps for ribosomal structure binding. All right. Introns. They are present in archaea and eukarya. Okay, they're not found in bacteria. Right. Antibiotics. They don't work against archaea and eukarya. Right? Histones are present in some species of archaea. Right? Circular chromosomes. Right? And then you end up, this is the real defining difference. Growth in extreme environments. All right? Most of the time, archaea have evolved in such ways that they allow for growth in very extreme environments. Okay? And part of that adaptations is seen in their characteristics. The difference in their cell walls. The difference in the membrane lipids. Okay? They allow for the cell to survive in these extreme environments. All right? They're enzymes that reproduce DNA and undergo metabolic functions have been altered in such a way so that they can survive in high temperatures, in high salt environments, low pHs, where other proteins are denatured, they're not. Okay? And that's part of their characteristics, which is directly related to their environmental habitats. So some archaea, most archaea, are what we call extremophiles. They live and thrive in extreme environments. Okay? So you have extreme halophiles. Halo refers to halogen, right? Chlorine is a halogen, right? That period of the table, of the periodic table, right? Halogen, chlorine, fluorine, iodine, right? So they like high salt or saline environments, right? Whereas extreme thermophiles like extremely hot environments, such as hot springs. Okay, the discovery of bacteria, or what we thought were bacteria, are bacteria in archaea that live in habitat hot springs where temperature is normally 85 to 100 degrees Celsius, and they live and thrive in that environment, has allowed us to do several different types of molecular biology techniques, utilizing the structure of their enzymes to do other stuff, right, such as polymerase chain reactions. All right, methanogens, right, such are archaea that produce methane as a waste product. All right, they are strict anaerobes and are poisoned by oxygen. So is that considered extreme environment? Right, methane is usually toxic to cells. Okay, and so you have swamps and marshes. Right, that's that distinctive swampy smell. And you drive by, the spring with ice melting now, and you get close down to Sharps Pond over here and Lake Massasoit. That's the decaying stench that is there. Those are bacteria and archaea that are producing methane. All right, so guts of cattle, okay, and deep sea hydrothermal vents are all archaea and bacteria that produce methane or that are methane. Okay, and so with that, what role do prokaryotes play in the world, in the biosphere? Right? The authors of the book state that prokaryotes are so important that if they were to disappear, the prospects for any other life surviving would be dim. If you think about it, when it comes down to the basic point, we really depend upon them in prokaryotes for several aspects of life. All right? In your gut, you don't produce B-complex vitamins. They are produced through the bacteria that reside in your gut, okay, in your large intestine, where they're breaking things down that you intake, and they're giving you B-complex vitamins in return, all right, as an example. Chemical recycling. All right, so we've talked about methanogens as being decaying, broken down material. What's those bacteria and archaea are breaking things down and returning what to the environment? Not just methane, but they're breaking, let's say, we're driving down 91 out here and I hit a squirrel. I just couldn't stop, okay? What's going to happen to that squirrel? It's going to decompose. That squirrel, which is a complex organism, protein, carbohydrates, lipids, in different structures of bone, muscle, fat, right? Brain, that's all going to decompose and eventually return to the environment in what? In the form of what? Carbon, nitrogen, phosphorus. How does it get there? Through the work of prokaryotes. Okay? So all the stuff that we put together, right, we ingest, we break down, and we put it together in our bodies as macromolecules, has to be broken back into smaller molecules so they can return to the environment in terms of the chemical structures. All right? So you have your chemoheterotrophic prokaryotes, they function as decomposers, right? They're gonna break down dead organisms and waste products, and return, their waste products are going to end up becoming what we use in terms of other stuff, all right? You could talk about water reclamation or reclamation projects. That's all based upon prokaryotic organisms, all right? So you can increase the availability of nitrogen, phosphorus, and potassium for plant growth. Right about now is the time where most, probably your parents, are starting gardens, okay? For me, in the fall, I take all the leaves that I collect that fall off the 2 huge silver maples I have in my backyard. I pull them all together, and I run them over with the lawnmower so they're really small, and I dump them in the garden. I leave them there over the winter, and they start to decompose. Okay? In the spring, they're not all decomposed. There's still plenty of leaf chunks there. Okay? But what I'll add to that is a little bit of nitrogen and bacteria. There's a little horse farm down probably just over the border in New York that gives away free manure. What's in horse manure? Nitrogen, and what else? Prokaryotes. Okay? There's gonna be a bunch of bacteria in there that are there to break down nitrogen. And what are they going to break down that horses eat? Hay, organic matter. I'm gonna take that, bring that home, spread that on the garden. They're gonna break down. They're gonna be happy because all this organic matter is there for them. What are they gonna return in the soil? Carbon, nitrogen, phosphorus for my plants to grow. Okay? So it's a matter of turning things over. And I do this every year, and it's been fantastic. All right, you can talk to Dr. Keys, who can go through she's retiring, but if you're interested in this, you can go through the sustainability project student group, and she runs a little bit of a seminar. I would call it a seminar series, or at least a workshop on organic farming. She runs a farm. She lives on a farm. She runs it completely organic. All right. All right. And then also, opposite to that, prokaryotes can also immobilize or decrease the availability of nutrients. Right. So they can actually store certain things inside their cells that no longer are available. Right. And we use this in terms of bioremediation, so that some prokaryotes can actually store lipids in the cells and not make it available outside, especially toxins. All right, we use this for cleaning up oil spills, like gas leaks. All right, so this is just a graph showing what? Uptake of potassium by plants, right? And then go through that. What are we talking about? These are ecological interactions, which are a symbiotic relationship between the plants and the prokaryotes, in a way. Okay? In some cases, there are bacteria that live on the roots of certain plants. And the root and the plant needs that bacteria, and the bacteria need that plant for survival. Okay? Rhizobacterium is an example. So the plant serves as a host, the bacteria is the symbiont, and they are working together. Right? One is giving substances to the bacteria is giving substances to the plant. All right? And this is often found between prokaryotes and larger organisms. The relationship, a lot of times, is mutualistic, whereby both organisms benefit, such as the bacteria, or such as the plant and the rhizobacterium. All right? Commensalism is another sort of a different take on it, whereby one organism benefits while neither harming nor helping the other in any significant way. All right? So it's there, but it's not doing anything for me, but he's not bothering me either, type thing. All right? And then you have parasitism. We all know what a parasite is, right? harms but does not kill its host. And then parasites that cause disease are called pathogens. Give me an example of a prokaryote that is parasitic. What causes Lyme disease? How do you get Lyme disease? Tick bite. What causes Lyme disease, though? Right, the bacteria, the spirochete that's transferred from the tick to the human. Okay, is Lyme disease necessarily fatal? Not really, not unless it goes on for a long, long time. Okay, so that's a parasitic relationship. Right, they're actually causing harm to the host in terms of over long-term neurological defects, rheumatoid arthritis. All right, and the spirochete benefits, has a nice, happy place to live, plenty of nutrients around, etc. All right, and I don't know why that was in there. Oh, this is an example of a ecological interaction. There are bacteria that live on the scales of this fish. All right, allow for luminescence. You see there. All right, but they also clean and maintain the structure. All right, the outside, so that the fish, whatever algae are on there, aren't overtaking. Okay. The communities of hydrothermal vents. What is a hydrothermal vent? Yes, sort of like a geyser, but it's usually in the deep part of the ocean. All right, so deep breaks in the ocean surface and the ocean floor. Right, there's an expansion of lava. All right, and you have this hot water, right, geysers, right, and then you end the vent, that sort of community of the fish that live in that environment, the plants that live in that environment, right, they depend on the chemoautotrophic bacteria for energy. All right, so the plants, fish, filter feed off that prokaryotes that are there, and all right, impacts on humans. When I talk about bacteria, what do you automatically think of? Bad, right? They're pathogens. You're going to get sick. All right. So most of the best known pathogens, right, are best known prokaryotes are pathogenic because that's the one we talk about. All right, but they have plenty of positive. All right, you have mutualistic bacteria in terms of the 5 to 1,000 species of bacteria that live in your gut, giving you B complex vitamins, helping you, absorb fat-soluble vitamins such as A, D, E, and K, etc. They break down food that is usually undigested by our small intestine, right? So then they produce these vitamins for us, other stuff, right? They also produce methane sometimes. Okay, but that's a mutualism relationship. Pathogenic bacteria, prokaryotes cause about half of all human diseases. About half. All right, Lyme disease caused by a bacterium carried by ticks, as an example. All right, and you went through this in lab. Most of you had to do a presentation, right? Based upon a disease that's caused by a bacteria, right? All right, so I don't have to beat that dead horse. All right, here's your spirochete. All right, the characteristic circular rash around the tick bite. All right, an example, there's a deer tick as the carrier. All right, pathogenic prokaryotes typically cause disease by releasing exotoxins or endotoxins. All right, the difference being an exotoxin is secreted by the prokaryote and cause disease, even if the prokaryote, all right, that produce them are not present. All right, so if you use like cholera toxin, what is cholera? Cholera is a type of bacteria. What does it do? causes inflammation and problems. The toxin, which is caused by the bacteria, right, has been in third world cultures is found in the water, right, and that toxin can cause intestinal discomfort, shedding of the lining of the intestine, and causes extreme diarrhea. Right, it's cholera. All right, endotoxins are a different type of poison, if you will. Okay, they are actually part of the bacteria itself. All right, so when the bacteria die, parts of the cell wall are released, and that parts of the cell wall is then toxic to cells. So it's endo, it's part of the bacteria. Exo, it's secreted from the bacteria, and the bacteria don't have to be present for that. All right, so it could just be a toxin that's in some other part. All right, so endo versus. Okay, we talked about horizontal gene transfer. Okay, and then you can, whereby one disease-causing strain can transfer the gene to a non-disease-causing strain, right? Which we talked about when we talked about transformation earlier in this. All right, in research and technology, all right, we use prokaryotes in the lab for DNA cloning all the time, to transfer genes to cells, to study how genes function in cells, all right, to replicate genes, all right, that's, E. coli is like used all the time in the lab, all right, and then Agrobacterium tumefaciens is used as a bacteria that's used to produce transgenic plants. All right, in the lab, we use bacteria to produce antibiotics, to produce insulin, to produce drugs and pharmaceuticals. All right, outside of it's sort of in a factory rather than in the. All right, and so bacteria can also be used to make natural plastics. Right, they're the principal agents in what we call bioremediation, right, the use of organisms to remove pollutants from the environment, and you can engineer them and their genes to produce vitamins, antibiotics, and hormones, and also produce ethanol from waste biomass, right, bioethanol. Questions on prokaryotes? Okay. With that, you'll have a quiz on that on Friday.